Good morning, children. Welcome back to online class today. Children, in the previous class, we all learned about the types of cell division. Isn't it, Mom? That there are three types of cell division that occur in animal cells. They are amitosis, mitosis, and meiosis. Mitosis is also called direct cell division. Mitosis is also called indirect cell division, and meiosis is also called reduction division. Yes, this amitosis is the simplest type of cell division occur in unicellular organisms. Then mitosis, there are two important events in mitosis. Who discovered mitosis? Fleming, isn't it, Mom? Now, mitosis consists of two important events. One is karyokinesis, another one is cytokinesis. This karyokinesis consists of four phases. We learned already what are the four phases. The four phases are prophase, metaphase, anaphase, and telophase. Okay. Now, you must know what is centromere. This centromere, or it is termed as kinetochore, or it is termed as primary constriction. Each chromosome is having a central body and an arm. This is called centromere. Centromere or kinetic core. This is the chromatid. Okay. Now this centromere is also called kinetochore. This chiasma. You must know what is chiasma. The point of contact, chiasma. The point of contact and interchange between chromatids of two homologous chromosomes. Homologous in the sense paired chromosomes. Okay. Then you must know what is diploid. Cells having two complete sets of chromosomes. That is diploid. It can be designated as 2n. Diploid. What is haploid? The cells having the only one set or single set of chromosome is called haploid. Okay, that is designated as N. This is haploid. This is diploid. Okay, children. So diploid means cells having two complete sets of chromosome. Haploid means cells having a single set of chromosome. You know what are chromosomes, isn't it, Mom? Chromosomes are the physical carriers of genes. Genes are the characters. Genes are the characters which are inherited or transmitted from parents to individuals. Okay. You know what is karyokinesis and cytokinesis, isn't it? What is karyokinesis? Division of nucleus into two. What is cytokinesis? Division of cytoplasm is cytokinesis. Karyo means nucleus, kinesis means division. So, karyokinesis means division of nucleus. Cyto means cytoplasm. Kinesis means division. So, cytokinesis is the division of cytoplasm. Today, we are going to learn about the third type of division. First two we all already learned. That is mitosis and amitosis. Then, the third type of cell division. What is it? Is meiosis. Isn't it? This third type of cell division that occur in animal cell, that is meiosis. Meiosis, it is also called reduction division. Reduction division. Do you know what is cell division? Division of cell. Division of cell is called cell division. So, the term meiosis, it is also called reduction division. The term meiosis was coined by Farmer. The term meiosis was coined by Farmer in 1905. Okay, this type of cell division produces sex cells or gametes. This type of cell division produces sex cells or gametes. Then why it is termed as reduction division? The chromosome number in the daughter cell is just half of the parent cell. Why it is reduction division? The chromosome number in the daughter cell is just half of the parent cell. So it is also called reduction division. Why mitosis is called equational division? The number of chromosomes present in the daughter cell is same as present in the parent cell. So it is called equational division. Okay, mom? Now, this meiosis, the term was coined by Farmer in 1905. This type of cell division produces sex cells or gametes. It's also called reduction division. You know why it is called reduction division. The chromosome number in the daughter cell is just half of the parent cell. So, it is also called reduction division. The chromosome number is reduced to haploid. Haploid means N, isn't it? So the chromosome number is reduced to haploid from diploid. Diploid it is designated as 2N. Then meiosis produces 4 daughter cells from a parent cell. So how many daughter cells are produced from meiosis? 4. 4 daughter cells are formed from a parent cell. It consists of 2 divisions. Meiotic division consists of 2 divisions. They are heterotypic division, then homeotypic division and heterotypic division. Meiosis cell division consists of 2 divisions, heterotypic and homotypic. Heterotypic and homotypic. Okay, these 2 divisions are there in meiosis cell division. Homotypic, it is also called first meiotic division. First meiotic division. Homotype is also called second meiotic cell division. Okay, mom? So, for first meiotic cell division is also called heterotypic. Second meiotic division is also called homotypic. Now, the first heterotypic or first meiotic division, it divides the diploid cell into 2 haploid cells. How many daughter cells are formed from meiosis? 4. The first meiotic division, from first meiotic division, the 2 haploid cells are formed from the diploid cell. Diploid cell can be designated as 2N. Haploid can be designated as N. So, first division, that is first meiotic or heterotypic division, it divides the diploid cell into 2 haploid cells. So, the daughter cells resulting from this division are differentiated from the parent cell in the chromosome number. Parent cell is having diploid set, but daughter cell is having the haploid set number. Okay. Then first meiotic division or this heterotypic division consists of 5 stages. One is prophase 1. First one is prophase 1. Second is metaphase 1. Third is anaphase 1. And fourth is telophase 1. These are the 4 stages. And one more, after this telophase, there will be the division, that is cytokinesis. So, this heterotypic division or first meiotic division consists of these following stages: prophase 1, metaphase 1, anaphase 1, telophase 1, and it is followed by cytokinesis. First, what is the first phase? Prophase 1. This is the first phase. Okay, first phase is prophase 1. This heterotypic division, the first phase is prophase 1. It's a longer phase. Okay, it takes place, it takes a longer duration, and it is subdivided into 5 stages. First stage is leptotene. Second stage is zygotene. Then third is pachytene. Then fourth is diplotene. And the last is diakinesis. These are the stages of prophase 1. What are the stages of prophase 1? Leptotene, zygotene, pachytene, diplotene, and diakinesis. Okay, what is the first phase under prophase 1? Leptotene. Second is zygotene. Third is pachytene. Fourth is diplotene, and the last one is diakinesis. Okay, these are the sub-stages of prophase 1. So, prophase 1, the first phase is diplotene. In this stage, during diplotene, the chromosomes become uncoiled. Okay, before that there will be the interphase. You all know this is the resting phase of the nucleus. Interphase is the resting phase of the nucleus. This is the phase between the two successive cell division. Next, prophase 1. You all know prophase 1 consists of 5 substages: leptotene, zygotene, pachytene, diplotene, and diakinesis. During leptotene, the chromosomes become uncoiled and assume long thread-like structure and take up a specific orientation inside the nucleus. Okay, they form the bucket stage. They form the bucket stage. The chromosomes forms the bucket stage. Now, second stage. What is the second stage? Zygotene. Zygon means adjoining. So, in this phase, 2 homologous chromosomes. What is homologous chromosomes? The paired chromosomes. 2 homologous chromosomes approach each other and begin to pair. So, the 2 homologous chromosomes approach each other. Homologous means the paired chromosomes. You know what is this? This is kinetochore or primary constriction, or it is also called centromere. The arms are termed as chromatids. Now, the 2 homologous chromosomes during zygotene stage, the 2 homologous chromosomes, homologous means the paired chromosomes, approach each other and begin to pair. The pairing of homologous chromosome is called synapsis. What is synapsis? The pairing of homologous chromosome. Homologous means the paired chromosome is called synapsis. Okay, what is synapsis? Pairing of homologous chromosome is called synapsis. So then what is the next phase? That is pachytene. Okay, so during pacus means thick. Okay, pachytene. Pacus means thick. So pachytene stage, the chromosomes are visible as long paired twisted threads. These pairs are called bivalents. Bi means two, isn't it? Bivalents. The paired chromosomes are called bivalents. Now each bivalent contains Each bivalent contain 4 chromatids. 1, 2, 3, 4. Each bivalent contain 4 chromatids. 4 chromatids. So it is also called tetrad stage. Tetrad means 4, isn't it? It is also called tetrad stage. In this stage, the chromosomes contain 4 chromatids. So it is also called tetrads. Then the homologous chromosomes of each pair begin to separate, but begin to separate. They do not completely separate, but remains attached together at one or more points by X-shaped arrangement. Here there will be the centromere, here the arms, chromosomal arms, that is chromatids. So X-shaped arrangement. Okay, but the the centromere do not completely separate but remain attached together at one or more points by X-shaped arrangement. This arrangement is known as chiasmata. Chiasmata. Singular chiasma, plural form chiasmata. So packaging stage, package means thick. It's very important stage. Okay, packaging. Package means thick. During this phase, the chromosomes are visible as long paired structures. Then the pairs are called bivalents. Bivalents. Bi means two. The pairs contain 2 sets of chromatids. So it is bivalents. The bivalent contain, see here, the bivalent contain 4 chromatids. 1, 2, 3, 4. 4 chromatids. So it is also called tetrad stage. Tetrad. Tetrad means 4. Tetra means 4. So it is also called tetrad stage. In this stage, the homologous chromosomes of each pair begin to separate. They do not completely begin to separate. So they do not completely separate but remain attached together at one or more points by a X-shaped arrangement. That is called the X-shaped arrangement of chromosomes, homologous chromosomes. This is called chiasmata. Okay. This next, what about the chromatids? Or what about the next stage? That is diplotin. Diplotin stage. In this stage, the chromatids break these points, or this chiasmata points, and the broken segments can be interchanged. Okay, these broken segments can be interchanged and and it is called crossing over. The interchanging of the chromosomal segments is crossing over. Crossing over. The interchanging of chromosomal segments. The chromatids break in these points, chiasmata, and the chromosomal segments get interchanged. It is also called crossing over. The process of interchanging of chromosomal segment is also called crossing over. So next is, crossing over occurs during packaging stage. So this stage is very important because crossing over and formation of bivalents and tetrads takes place in this stage, packaging stage. As a result, the genetic recombination takes place. Because of the interchanging of chromosomal segments, the genetic recombination takes place. What about the next phase? That is diplotin stage. During diplotin stage, each individual chromosome of each bivalent begin to separate longitudinally into 2 similar chromatids. Now, the homologous chromosomes repel each other and separate. The chiasmata begin to move from the centromere towards the end. Okay, resulting in terminalization. because it will move towards the end, that is terminalization. So during diplotin stage, what happens? Each individual chromosome of bivalent begin to split longitudinally into two similar chromatids. Now the homologous chromosome repel each other and separate. Now the chiasmata begins to move from the centromere towards the end. This process is called terminalization. Then what about the next is the diakinesis. The next one is diakinesis. The paired chromosomes now shortened, thickened, and new nuclear membrane, and now nuclear membrane and nucleolus begin to disappear. This during diakinesis, this phase, the paired chromosomes are shortened and thickened, the nuclear membrane and nucleolus start to disappear. Spindle fibers make their appearance. Now, so during diakinesis phase, the nuclear membrane and nucleolus start to disappear. Spindle fibers start to form inside the cell. Okay. Next, what is the next phase? Is metaphase 1. Okay. Next is the second phase is the metaphase 1. So I already told you prophase is 1 is the longest phase. It includes 5 stages: lactotin, zygotin, pachytin, diplotin and diakinesis. Pachytin stage is very important because it this chiasmata formation, then chiasmata formation and genetic recombination occurs in this phase. So it is very important. Okay, next phase is the metaphase 1. In metaphase 1, the chromosome move towards the equator. The chromosomes move or lie at the equator. Okay, move towards the equatorial plate and the centromere does not divide. During metaphase 1, the centromere or the central body of the chromosome does not divide. Then what about the next phase? Anaphase 1, isn't it? Anaphase 1. In this phase, each homologous chromosome with its two chromatids and undivided centromere move towards the opposite pole. Okay, during anaphase, the homologous chromosomes with the two chromatids and the undivided centromere move towards the opposite poles. This stage is called dyad because the chromosome is the centromere with the two arms. It is also called dyad. Okay. Next, what is the next stage? Is the telophase 1. After reaching the respective poles, I already told you in this phase, which is the metaphase, the chromosomes lie at the equatorial plane. Anaphase, it move towards the opposite poles. Here, the telophase 1, after reaching their respective poles, these chromosomes become uncoiled. Uncoiled, the nuclear membrane nucleolus start to appear inside the cell. Okay, nucleolus start to appear inside the cell. Then what is the next phase? It is for then telophase. In this telophase, two daughter cells are formed, isn't it, mom? Two daughter cells are formed by telophase. Then this telophase is followed by what's the next one? Cytokinesis. Cytokinesis means division of cytoplasm. Now cytoplasm is divided into two cells. Okay, cytoplasm, this division of cytoplasm results in two diploid daughter cells. Okay, now thus by first division, heterotypic division, two daughter cells are formed. By heterotypic division, two daughter cells are formed. Okay, then second type of division which occur in meiosis, that is the homotypic division. We can learn it in the next class. Okay, children, listen. So meiosis cell division is also called reduction division. It includes the certain phases. What are they? Prophase 1, metaphase 1, anaphase 1, telophase 1, that is followed by cytokinesis. Cytokinesis, division of cytoplasm. This prophase 1 is the longest phase. That includes 5 phases: leptotene, zygotene, pachytene, diplotene and diakinesis. Among all these phases, a genetic recombination takes place in pachytene stage. Crossing over and formation of chiasma takes place during pachytene stage. Okay. So pachytene stage is important. Next, metaphase stage, chromosomes lie at the equatorial plane. Anaphase the chromosomes with our bivalence move towards the opposite poles. Then telophase 1, the nuclear membrane and nucleolus start to appear. That is followed by cytokinesis. Thus, two individuals are formed by this mitotic, this mitotic first division or heterotypic division. Children, now you all turn page 221. Under the heading heterotypic division, you are writing what are the stages of heterotypic division. You have to mark 5 stages: prophase 1, anaphase 1, metaphase 1, telophase 1 and cytokinesis. Next question, near the heading prophase 1, you are writing name the stages of prophase 1. Name the stages of prophase 1. You are marking the third line: leptotene, zygotene, pachytene, diplotene and diakinesis. Then under the heading zygotene, you are writing what is synapsis. What is synapsis? Pairing of homologous chromosome is called as synapsis. Okay, children, you are writing these 3 answers in the learning and writing these 3 answers in the notebook. Thank you, children. We shall meet again in the next class.